Hello, everyone. Welcome to the webinar on cancermodels.org, which is an open catalog for patient-derived cancer models. My name is Tushar Mandloi, and I'm a bioinformatician, part of the phenomics team. And I've been working on this project for more than a year now. And I mostly work with the data, which goes into the platform, which we'll be having a look at. So in today's webinar, we'll first have a look at what are patient-derived cancer models, followed by an overview of cancermodels.org, where I will talk about the models and the data, the feature of the resource, also give you some demos on how you can use it. And next, we will have a look at what are the next developments we are working on. And lastly, how you could get involved with us. Before we start, let's have a look at the objectives of today's presentation. After the presentation, you will be able to search for the patient-derived cancer models of your interest. You'll be able to explore their associated molecular data, identify potential collaborators who generate these models, and understand the quality representation of the PDCMs within the platform. So cancer is still leading cause of death worldwide, accounting for nearly 10 million deaths in 2020. With most common cancers being breast, lung, colon, and prostate cancer. And the advancement in cancer research, early detection and treatment have led to improvement in cancer-related outcomes over time. However, it still remains a significant global health challenge. Cancer is a complex heterogeneous disease that can vary significantly between patients. And to better understand and study cancer, researchers utilize various patient-derived cancer models. So let's have a look what are the patient-derived cancer models. So these models aim to capture characteristics and diversity of tumor observed in patients. The PDCMs are majorly categorized into three categories, such as PDX, PDC or 2D cancer models, or patient PDOs or 3D cancer models. What do you mean by PDX? So PDX are patient-derived xenografts. These models involve implanting the patient tumor tissue directly into an immunodeficient mice. These models retain the genetic and histological features of the original tumor and can be used to study the tumor growth, metastasis, and response to therapies. PDX models are particularly valuable for evaluating the efficiency of anti-cancer drugs and investigating the tumor heterogeneity. Next PDCM is PDOs, or patient-derived organoid models. These are three-dimensional cultures derived from the patient tumor tissues or healthy tissues. They can mimic the architecture of the original tumor, including multiple cell types and genetic alterations. PDOs are used for studying cancer biology, drug screening, personalized medicine, and investigating the effects of tumor microenvironment on treatment response. And lastly, the last category is the PDCs, patient-derived cell lines, or 2D models. PDCs are established by culturing tumor cells, often directly from patient samples. These cell lines are used for in vitro studies to investigate tumor biology, genetic alteration, and signaling pathways. PDCs are valuable tools for high-throughput drug screening and mechanistic studies. So each of these PDCM has their own strengths and limitations, and researchers often use a combination of models to gain a comprehensive understanding of tumor biology. Some of the key benefits of these models are preclinical drug testing, recapitulation of tumor heterogeneity, predictive biomarker discovery, or personalized medicine. These models also provide value in various sectors such as academia, clinical, and pharmaceuticals. And since now we have understood the value of these models, you must be wondering, where can you find them? So there are various resources and databases out there that specialize in cancer research and genomics, which provide you with model information and their molecular data, such as PDMR, GDC, and consortiums such as PDXNet, Pivot, or individual laboratory which generate these models, such as Jackson Laboratory or Charles River. But since there are various resources, that comes with their own set of challenges. So existence of these multiple resource and databases, the challenges which you would face is that each resource has specialized in different cancer types. They adopt their own different data formats, analysis pipelines, and quality control measures. And each resource would have their own infrastructure and curation process. And due to lack of a standard, it leads to heterogeneity in data models and difficulty in data integration. And to deal with these data-related challenges, there have been initiatives to handle these heterogeneity, one of them being FAIR data principles, which stands for findable, accessible, interoperable, and reusable. These practices fosters collaboration, encourage data sharing, facilitate the development of tools and platforms that support seamless data integration and analysis across different data sets and sources. What does it mean to be findable? It means that you can look for it over the internet, you can have a link and you can find it. Accessible means you can access that data using a link or a URI. What it means to be interoperable is that you can integrate different data types into your analysis pipeline, and reusable meaning that you can rerun those pipeline over time and you will still get the similar or same output. The resource we will be seeing today is built upon the success of a previous project, which was supported by ITCR. And in 2016, our team coordinated with the community in creating a standard for PDX minimal information standard by adhering to the FAIR data principles, which we looked in the previous slide. So the standard consists of 4 modules: clinical, model creation, model quality assurance, and model study or associated metadata. These modules have been put together with input from 35 research institutions and organizations, and PDXMI has become the established in the community for data exchange, adopted by the model providers, consortia, and informatic tools for integrating PDX data. And development of this standard led to a cornerstone for the creation of a resource known as PDX Finder. With this resource is an aggregated and harmonized PDX models, has aggregated and harmonized PDX models, and their associated molecular data. We have recently retired PDX Finder, as we now have our own new resource called the cancermodels.org, where we expanded PDX Finder to incorporate different PDCMs. With support from ITCR, we developed a new resource while adhering to the FAIR data principles we had a look a few slides back. This helped us in improving our data model to make data findable and accessible. With help of the standardization, we ensured that there's interoperability and reusability of the data. Our prototype resource was called PDCM Finder in 2021, where we included 2D and 3D cancer models. And along with the feedback from our users, we worked on our minimum viable product, which we named as patient-derived cancer models. And now in 2023, we have our product, which is called as cancermodels.org, about which you are here to learn about. As of now, we have a total of 33 data providers from across the globe such as Jackson Laboratory, Sanger Institute, Broad Institute, etc. And majority of our providers are also part of consortia such as PDXnet, Pivot, EuroPDX, etc. In our resource, we have more than 7,000 cancer models with around 5,000 PDX models, 1,500 cell lines, and more than 350 organoid models. We also cover various cancer systems and have some rare cancers like head and neck cancer, and we are constantly working on incorporating more PDCMs in our resource. Out of the total models, there are 17% of the models from pediatric samples and 83 from adult samples. And here is a bit of an overview of the data available in our resource. We collect various molecular data types such as gene mutation, copy number alteration, expression, etc. And we are working on including more data types associated with these models as well. We provide the users with such facets for easy navigation and exploration, leading to improved findability and accessibility of the model of their interest. We will have a look at these filters in upcoming slides when I'll give you a demo on how to use the resource. User can filter models based on their type, dataset available, tumor type, or patient sex, etc. Another feature is that we harmonize the metadata for easier comparison across models, and this makes the data interoperable and reusable. So now let's have a look at some of the use cases for better understanding of how you can use the resource. So let's take an example that Kelly is an oncology researcher who's looking for triple-negative breast cancer models with recurrent tumor type. So Kelly So Kelly visits cancermodels.org. She clicks on the search page for the models of her interest. Then she makes use of the filter provided on the left side and filters on the patient tumor as recurrent. Then she selects the cancer system as breast cancer, followed by applying a filter on available breast cancer biomarkers. Then she selects the estrogen negative, Herceptin negative, and progesterone negative models. And after applying these filters, she finds there are two models in our resource which fits in her criteria. Model 1 has various molecular data available as compared to the model 2. You can see the metadata associated with the models in the middle section. We will have a look at the model characterization bar in upcoming slides. And then Kelly selects the first model as her model of interest. She can now view the model information in detail, such as cancer stage, patient ethnicity. She is interested in the biomarker information, so she heads over to the molecular data section. She clicks the view data button for cytogenetics data type, where you can see that the biomarkers are triple negative. We also integrate with external resources, which can provide further information about these genetic markers. The external resources we connect with are Civic, OncoDB or OncoMX, dbGaP, dbSNP, etc. Here you can read about the markers. Then we also provide information about the patient treatment associated with the models, and also provide information on publications with these models associated with. So let's have a look at another example where Holly, another researcher, is looking for skin cancer models from treatment-naive patient with mutation in BRAF gene. So Holly visits cancermodels.org. Then she visits the search page. She then filters on model type as PDX models. Then she filters on the patient tumor section. She filters on the cancer system as skin cancer. Then next, she filters on gene mutations. She selects the models with gene mutation in BRAF gene by typing in the gene symbol. Next, she filters on the treatment status as treatment naive. And by applying these filters, she's presented with 9 models of her interest. And we can see the model information and the available associated genomic data with the models. She selects the model of her interest, where she's presented with the metadata associated with the model, the model engraftment data, and model quality control information. She's interested in the molecular data. She heads over to the section. Then she clicks the view data button for mutation data, where she can filter on the gene symbol of her interest. In this case, is BRAF. She can now see the amino acid chain, the consequence of the mutation, allele frequency, etc. She can read more about the gene by clicking on external resources we integrate with. She can also look into the variation by going to dbSNP, where you can find out the position of the mutation and other associated details. You can also download this data by clicking the download data button, in which you'll get like a CSV file with all the mutations associated with the model. We also provide drug dosing information, which drugs were tested and what were the response. And if she would like to have a deeper look or purchase this model, she can visit the provider page by clicking the view data provider. In this case, it's the Jackson Laboratory. So you will be heading over to the Jackson Laboratory, where they show the model information and additional information, such as histology of the model. And let's see another use case where Sam, another oncology researcher, looking for reproductive system cancer cell lines from female patients. So Sam with his cancer model heads over to the search page. Then he filters on the model type as cell lines. Then he selects a model from the female patients. And he also applies the filter on the cancer system as reproductive cancer, as he is interested in models from reproductive system. He is presented with the model of his interest. The models which apply based on the filters he applied, and there are 123 models based on his search. He picks the model of his interest. He is presented with the metadata and molecular data of the model. Since he is interested in the expression data, he heads over to the molecular data section and selects the view data button for the expression data, where you can see we have information on we have FPKM value, TPM value, and counts. You can also fetch the raw data by clicking the raw data link for performing further analysis. For raw data link, we integrate with EGA and ENA. He can also view the data or more information about the model at the provider side by clicking View Data at Provider. So now let's get back to the model characterization score, which we saw in the demo. So we use it as a metric to showcase the available model information. Here we can see that model 1 and model 3 have a richer bar compared to the model 2. These scores are computed based on the available information in the resource for the models. So if we compare model 1 with model 2 in detail, we can see that model 2 has a lot of fields with missing information, such as if you see patient age for model 1, it falls under 70 to 79, but for model 2, it's not provided to us. Same goes for cancer grade, cancer stage, patient ethnicity, and collection site. So, sorry about that. So that's why you'll see model 2 having model 1 having a richer model characterization score bar compared to model 2. Lastly, let's have a look at various molecular data associated with these PDCMs. And so you can explore these PDCMs also via our data overview page, where we make use of various interactive data representation. Here we categorize the models based on the primary site of tumor, and we select pancreas as the primary site. We can see there are 333 results with primary site as pancreas. And now we have a look at what molecular data we have for these models. We got mutation, copy number, alteration, and expression data here. For copy number alteration, we have log2 CN ratio. For expression data, we have FPKM, TPM, and Z-score values. These along with the drug dosing information associated with the models. Lastly, I would like to bring this session towards the end by talking about our next development. We are working on integrating various immune-related features, curation of rare models for maximizing their utility, improving our data submission process for enhanced data sharing. We also work on new data visualization and integration of resources for maximizing the model value. We will be expanding our model quality rating for improved reusability. You can get involved with us by filling in the minimal information standard for 3D cancer models, or you can provide us your feedback about the resource by filling in the user survey. And if you would like to use the data, you can make use of our API. And you can find us at www.cancermodels.org, and you can also connect with us on Twitter. Lastly, I would like to thank Helen, our PI, Zina, our project lead, and my teammates, Mauricio, Federico, Marcelo, our collaborators at Jackson Lab, and our SAB board members and our providers in the development of this resource. I would also like to thank you for being patient with me. And yeah, are there any other questions?
Thanks a lot, Tushar, for today's webinar. And thank you everyone for joining in today. And now is the time you can type in your questions in the Q&A box if you have. So we have one question already in there. So the question is, does every model have different accuracy?
So I would say yes and no, because so not every provider provides us with information about the accuracy. So so then if you compare the models across different providers, there's chance that you won't be able to figure out like what's the if they have a different accuracy, even though they might have like similar. Let's say in case of PDX, they have similar mice that it was tested on. So I would say yes and no.
Next question is, are models free of charge? Are they available to pharma?
So we are an open non-profit resource. So you can find out information about the models, but then to obtain these models, you'll have to go to the individual provider's website, and then they might have their own charges, and also based for the availability to pharma, there not every provider is open for that yet. So some of the providers are available, some of the models are available to pharma, yes.
Thank you. Do you only work with curated data?
Yes, and if we need some, we also perform some of our own curation, and if we need some help, if we need some clarification, we get back to the model providers to make sure that the whatever we present on the portal to the user is curated information. Yeah.
Maybe I can ask a question. So, do you always get requests from research labs to upload the data on this platform, or you also go through the publications in terms of cancer research and you get data from there and you upload it on the website?
So for most of the models present in the resource are from the providers, from academic or industrial providers, industry. But we also we are working on like fetching the data from the publications directly as well.
Okay, so we have a new question here. You raised some research scenarios about a particular tumor. What if I want to bulk download massive data for analysis in deep learning?
So you can make use of our API, where you can fetch the molecular data or even the metadata of the model. And then if you would need some help we also have the documentation of the API, which was here. If you can find, I can share the link in the. If Mauricio, could you please share the link in the chat for the API, and then if you need some help, you can always reach out to us.
Okay, I think the next message is just a compliment for you. Thank you very much, Tushar, for an excellent presentation. I'm so impressed by how you communicated. You are such an incredible solution provider.
Thank you.
All right. So next question is, we have NCBI GEO database. If I want to compare the healthy versus diseased or cancerous cell, how is it different from GEO? I guess how your cancer models platform is different from GEO.
That is a really good question. I would say in GEO, right now we have information about the models from tumor patients, so you will not be able to compare on our platform directly with healthy samples, but you can then download this data and then make comparison with the disease, like healthy and diseased cells.
Okay, so one question has come in. Would you please show us the clue to use a cancer model along with medical images for cancer prediction?
So right now we haven't made the image data for the cancer models available on our resource yet, but it's work in progress. I can assure that. So on the platform, I'm not sure if this question cannot be answered right now with the current stage of the platform, but in future you'll be able to compare models also based on the medical images,